thanks Academy for the fellowship as well as inviting me to give a talk over here. I am basically chemist, and last probably 15 or more than 15 years I am trying to understand biology. So I am hardcore chemist. So today I am going to talk about one of the kind of research we are doing. So my lab mostly working on protein misfolding and aggregation, and how this aggregation lead to one kind of specialized structure called amyloid that is associated with almost many human diseases like Alzheimer's, Parkinson, type 2 diabetes, many diseases. And also we are working on that can we design, because these amyloids are kind of very robust and kind of stable material. In our lab we are trying to understand that if we design very nice this amyloid biomaterial, can we use for the different kind of biotechnological application like stem cell differentiation, drug delivery, or organoid modeling. So anyway, so today I am going to talk about one of the research we are doing last few years, that liquid-liquid phase separation of the proteins and polypeptide, and we propose that liquid-liquid phase separation or liquid condensate is one of the alternate state of the protein and polypeptide. So I think we all are aware of phase separation of particular molecules. Here you can see that how water phase separate and make gas, solid ice as well as liquid water. So also, I mean, we have seen that if we put the oil in water and stir, we can see that lot of liquid droplet emerge, and then they coils and make a large liquid droplet. Also lava lamp where you put a wax and you heat a little bit here, then wax goes up, again come down. And most often also that small liquid droplets, they do Oswald ripening, small droplet, they merge together and also make a larger kind of liquid droplet. So how this is relevant for the biology? I will say almost like 100 years back, Wilson proposed that the protoplasm of the cells actually many suspended drops, and these all drops probably do the specialized functions. That's the way probably cell works. However, really, although like many scientists studied the liquid-liquid phase separation, but new way it came in 2009, that when Brangren and their group studied that when C. elegans egg cells actually divide to the sperms and germ cell line, and there is a RNA binding, RNA containing the granules actually is separated in the posterior part of the cells. So people didn't understand that how it works. Then they proposed that it's liquid-liquid phase separation. And at the beginning, everywhere in the cells there is a liquid-liquid phase separation, but when it's actually divide, that is localized here due to the kind of specialized protein which kind of stabilize the liquid-liquid phase separation of the protein. So that's the way probably phase separation in biology started at around 2009, and you can see during time people discovered that many kind of membraneless organelles, they actually form due to liquid-liquid phase separation like P bodies, Carl bodies, nucleoli, PML bodies, stress granule, and they all are evolved for the native functions of the host organisms of the cells. But as you know that how actually these membraneless organelles evolve, because membrane covering with the organelles is very important for specialized functions. So people propose that there is a huge advantage for these membraneless organelles, because they can work like reaction crucibles, and basically cell can mix when they want and they can dissolve when they want. Basically there will be very nice regulation for a particular reactions for this membraneless organelle formation. So you can sequester any molecules whenever it's required. So then one of the example that if yeast, they form stress granule, they survived. If they don't form the stress granule, they actually die. So that indicates that these membraneless organelles, how important they are for the cells, and how cell actively regulate their formation and their dissolution for their functions. So of course there is a cost, because for these membraneless organelles formations, proteins molecule has to come together, and then they have to be concentrated, and therefore that there is a chance for protein misfolding. And that's the way it's working for many diseases. People propose TDP43, FUS and tau So they actually aggregates from the liquid-liquid phase separation. So we propose, because we are working on alpha-synuclein for many years associated with Parkinson's disease, we propose that the nucleation step for alpha-synuclein aggregation could be liquid-liquid phase separation. So first we tested with algorithms, and we do find they have low complexity domain as well as prion-like domain that is kind of essential for liquid-liquid phase separation. And you can see that when we put the crowders, we can see at high concentration and high peg, they actually do form liquid-liquid phase separation and liquid condensate formations happens. But if you decrease the pH, you can see that even in absence of peg, as low as 10 micromolar protein concentration, they form liquid-liquid phase separation. And you can see with FIDC level protein, you can see that how liquid condensate forms, but they are actually reversible at the beginning. That means that if you little bit heat, they dissolve to form a solution, but during time they get aged and they solidified, then you cannot make a kind of reversal. So then with time when we tested, we realized that these small liquid droplet, they actually when they touch, they merge, they make bigger droplet, and with time when we did the FRAP, you can see there will be very less recovery. You can see here that at the beginning there will be 100% FRAP recovery, but with time there will be very less recovery, suggesting they are actually undergoing liquid to solid transition. And we also tested with the anisotope PDK analysis, also this is support that there will be solidification with time. And here you can see that how liquid droplet they are forming, small droplet actually they are dissolving and they are forming larger size droplet with time. So then we ask the question that how this is relevant, this liquid-liquid phase separation with Parkinson's disease associated conditions or solidification or amyloid formations. So what we did, we tested different conditions like exposure of different metal ions, liposomes, as well as disease associated mutation, and we have seen that all these conditions, whatever it's promote protein aggregation, is actually indeed promote the liquid-liquid phase separations. And when we did the FRAP analysis, we did observe they actually not only promoting liquid-liquid phase separation, they are promoting the liquid to solid transition as well. And then we ask very important question, because this is very traditional car people observe for the protein aggregation field, Alzheimer's, Parkinson, whatever type 2 diabetes, whatever disease field people study, that these are traditional aggregation graph, nucleation dependent polymerization. So we ask questions where actually liquid-liquid phase separation arise. So then with time what we did, that we start the reactions, and every time we actually separate out and look at under microscope where liquid-liquid phase separation happenings and what kind of species the protein is generating. So basically we did very extensive work, and we were able to show that at the beginning of the lag phase there is a liquid-liquid phase separations, and here when nucleation, basically that fibril formation nucleation start, here there is a liquid to solid transitions happenings, and at the end you can see that there is a amyloid fibril formation emerging either from the surface or from the interior of the droplet. So we did also cellular study, and we were able to show that inside the cells alpha-synuclein also undergo liquid-liquid phase separations. Also with time they solidified, and we were able to show they form kind of amyloid kind of aggregates, and that's the way we propose that this could be one of the mechanisms that how alpha-synuclein aggregates to form amyloid fibers. So at the beginning it could be liquid-liquid phase separations, and with time that liquid to solid transition, we are able to also understand which domain of the alpha-synuclein actually playing role for the liquid-liquid phase separation as well as subsequent solid transitions. So then one of the major questions in the field, that protein has to be unstructured domain or prion-like domain to form liquid-liquid phase separation. So we ask, what about 80% proteome where most of the proteins are structured? So what we hypothesize, that actually all protein literally can form liquid condensate. How? So you can see here that unstructured protein can form folded proteins and it will be solvated with water. But if there is a water exclusion with crowding, or there is a large number of proteins interact each other, there will be water exclusion, and it can form liquid-liquid phase separation, and which eventually can transform either amyloid-like condensate or solid-like condensate, depending upon the protein sequence and experimental condition or cellular conditions. So to test this hypothesis that liquid-liquid phase separation could be genetic property of the protein and polypeptide, we choose 19 different proteins with different sequence, different secondary structure, and we tested all kind of algorithms. Some of the protein possess the liquid droplet propensity, some of the protein don't have intrinsic disorder domain, some protein do have. And then when you look at, we do observe all of these proteins, they do from liquid condensate, and however with different phase space. That means that different concentration or different crowding is required per individual protein. Then at the critical concentration, we set up that what is the kinetics of the liquid-liquid phase separation. We have seen that whichever protein have low critical concentration, they actually phase separating very fast. You can see here that critical concentration and T-half of the phase separation kinetics, you can see they are not exactly, we are not expecting exactly correlation because we use the different concentration here. We use the critical concentration of the protein for the phase separation kinetics. However, there is some rough correlation exist. So then we characterize all these proteins that with their Fab recovery, you can see that most of the protein, they are in liquid state. If you look at that, most of the protein they recovered almost 100% and very fast recovery. However, some of the protein do recover slowly or partial recovery happen. That means that they might undergo that some kind of solidification at the beginning. And you can see here that we calculate the T-half. You can see different protein have different T-half. Some protein have actually larger T-half, suggesting they might undergo partial solidification with time. So we checked their fusion. That means this is one of the property that liquid droplet, when they touch each other, they should fuse and make a larger droplet, and that is one of the property of the liquid. And we have observed that all the proteins, whatever droplet we observe, when they fuse, when they touch each other, they actually fuse and make a large droplet, and they're mostly reversibles, and they keep intact their secondary structure by circular dichroism, and this is the kind of electron microscope suggesting they are undergoing the phase separation. So one of the questions is asked in the field that whether using PEG we are artificially introducing the property to the protein and they are probably phase separated. So then what we did, we took the cellular extract, cytoplasmic extract, and when we studied, most of the protein actually do undergo phase separation in the cytoplasmic extract, suggesting this is the intrinsic property of the protein, and crowding is kind of essential for the phase separation. So then, of course, this is further we characterize with the FRAP, and also their fusion event, suggesting that in the cellular extract, all the protein do from liquid-liquid phase separations, undergoing fusions and making larger droplet. So then we ask that is the solidification property of the intrinsic property of liquid condensation of the proteins. So we do see that many protein they undergo solidification. That not essentially means that when protein solidify, they will lose the functions. So basically many protein when they solidify, they can be reversible and can do functions, again can come back the monomeric state. So we've seen that most of the protein with time, you can see that their recovery time is much higher. Some of the protein didn't recover at all, that means it's complete solidified with time. Here you can see there is also some of the protein, there is a huge structural change with solidification, some of the protein don't. However, only few protein you can see that they are showing thioflavin T. That means that these subset of the protein undergo amyloid formation with solidification, you can see with alpha-synuclein, but all other protein, they are normal droplet but with solid droplet. So then we ask the questions, what is the property of the protein sequence? How it is forming the liquid-liquid phase separations? We've seen that actually charge on the surface, folded protein, because most of the charge will be on the surface, right? The charge interactions playing a significant role. When we plot critical concentration of LLPS and their kind of net charge, we have seen there is two populations. One is higher molecular weight, another is lower molecular weight, and this is kind of expected because liquid-liquid phase separation also promoted by higher polymer length. And we have seen that whichever didn't fit very well, they actually have kind of exposed hydrophobic surface that is also supported by ANS fluorescence. And then we have seen that these proteins, they undergo both the ionic interactions and with exposed hydrophobic surface, because if we add kind of which block exposed hydrophobic surface, then like 1,6-hexane diol, actually they kind of removed the liquid-liquid phase separation property. And the protein which undergo liquid-liquid phase separation with, kind of ionic interactions, they actually gone, that property is gone when you add the salt. And then all protein we actually tested with light scattering, that what kind of interaction playing a role for liquid-liquid phase separation with all these 20 protein, we observe amazing things that some set of proteins we have very high kind of charge on the surface, they undergo through charge, whichever have exposed hydrophobic surface, they goes through exposed hydrophobic surface, hydrophobic interactions. Otherwise, many protein undergoing simple hydrogen bonding forming liquid condensate. And then when we study the interaction potential, we have seen that this phase separation tendency is directly proportional to their interaction strength with SPR. Very interesting, you see alpha-synuclein here. In absence of salt, 700 micromolar is the critical concentration. You just add physiological concentration of salt, 150 millimolar, let's say sodium chloride, you see 100 micromolar is the critical concentration, and you can see their KD also decreasing significantly. For example, GG, you see very nice interactions. In absence of salt, when you put the salt, you can see completely interactions demolished, and you can see no liquid-liquid phase separations. So I will skip that. So then we did for the with peptide, for example, lot of people propose that there is a sticker and spacer requirement. That means you need a spacer like a polyglycine, and then you need a sticker to stick the interpeptide or interprotein interaction for liquid-liquid phase separation, and we said probably it is not required at all. So then what we design? We design simply polyvaline, it will be complete sticker, glycine, complete spacer, and then polyarginine and polyaspartic acid. And you can see that glycine, you can see here that 2 millimolar concentration is from liquid-liquid phase separation. Valine, 1 millimolar is forming liquid-liquid phase separation. Obvious, because your hydrophobic interaction is much more stronger. And glycine, probably there is no other way, only hydrogen bonding playing a role. But these two peptides, in absence of salt, they don't undergo liquid-liquid phase separation, but if you add salt, they do form phase separations. And again, if you mix them, polyarginine and polyaspartic acid, they undergo phase separation. So with that, we also did kind of huge amount of study to show how multiphasic and heteromeric co-phase separation happens and how is selective for the protein. For example, some protein, they co-phase separate two protein, but if you look at here, some pair, they don't phase separate. Actually, they promote one protein for phase separation, but other protein, they actually demolish the phase separation. So basically, it's all about sequence and interaction strength and environment probably dictating the phase separation property of the protein. So where the field is going, so one of the things I think the future that one might even think artificial organelle formation in the cells, and for the drug discovery project, as well as function, native function of the cells, I think that could be the future for liquid-liquid phase separation. This is my group. Thank you very much.
So I was just wondering whether you can drive the liquid-liquid phase separation by inducing a stress condition. Like if you give a heat stress, do you think that there will be more proteins which are usually doesn't really go for LLPS, but they are now more prone to LLPS? And the second question is, do you think that LLPS helps in the protein stability, means the half-life of the proteins? Yeah.
It's a very fantastic question. So I have not shown the data for P53 actually. I mean, you know that how P53 works. So we showed that P53 condensate formation actually is for the they do native functions. And why? Because it's increased the stability and is resistance against proteases. So if you look at same concentration without phase separation and with phase separation, you can see different stability as well as protease resistance, and so that basically they are actually much more stable. So other question is that with heat shock actually in the cell, so for the here in vitro it will be probably difficult to replicate, but inside the cells they do form and they actually favor the phase separation, and that's the way probably cells survive, because that's the way they form the stress granule. Yeah, thank you.
Hi, Samir here.
Hi, hi.
Hi. Samir, very interesting talk. Just wanted to know that in these kind of phase-separated condensate, you sometimes also see certain nucleic acids.
Right,
So the nucleic acids in combination with protein.
right.
My question to you is, can you give me some insight that how the positioning is done? So for example, you have 2-3 different kind of proteins and a nucleic acid. So what comes to the core? And what forms the periphery? Because these are all membraneless organelle kind
Right.
of condensate.
Right.
So do you have any insight?
Yeah, so people probably propose that positive negative interactions they actually playing a role, because these condensate they actually loosely bound, because if they are very tightly bound there will be solidification, then lot of solidification will hamper for the cellular functions. So that means that you have a positive charge protein and negative charge DNA, they actually neutralize that charge based interactions and they are making a complex, so that there will be large kind of hydrogen bonding network or interaction network to form liquid condensation. So I can tell you in P53 context, so we seen that if you add specific DNA, so P53 liquid condensate they are dissolving. But if you add RNA and non-specific DNA, they are actually promoting phase separation. So it's very nice that how specific they are, although we don't know, because it's very difficult to do NMR in the phase separating condition. So when you add PEG, it disturbs a lot of NMR properties. So we have not done NMR, but we are able to see that how sequence of DNA also regulate their formation and their dissolution of the liquid condensate, and that's the way probably is regulate the function of the liquid condensate. Yeah.
Yeah. So some of the liquid phase separation you are following by light scattering, right?
Right.
But that could be just due to formation of smaller aggregates. It doesn't mean you have phase separation. And then also when you follow by fluorescence,
Yeah.
you are adding a hydrophobic dye, presumably for labeling, which can that also be promoting the
Very good questions. So the thing is that in the light scattering, of course, that if there is aggregation also it will show. But what we do that during light scattering we check. So every let's say 1 hour or 2 hours we check under microscope whether it is due to aggregation or liquid phase separation. So another thing is that your second questions. So that we use the 10%. So basically that we have 90% unlabeled proteins and we have 10% labeled protein we add. You can go as low as 1% labeled protein. And actually if you even don't label, you can see in the IC, but probably you cannot see. So at probe that it's undergo liquid phase separation, but probably you cannot see under the microscope. So this is kind of caveat trade-off you have to do, at least to label some percentage of the protein.
These low molecular weight heat shock proteins, which are associated with something like leprosy. Okay, is there any role of this liquid condensate formation?
So heat shock protein actually a lot of people are proposing, they actually kind of dissolve, they work like a anti-liquid phase separation where protein undergo liquid phase separation and amyloid formations. But I will not be surprised if in protein itself they form liquid condensate formation. So for example, now we are defining that this protein undergo liquid phase separation. How? By microscope, right? But if it is tiny, something like let's say 5 nanometer liquid droplet, how we can see? So there one can even hypothesize that every condition whatever you are doing, let's say protein, they probably micro condense it, we don't see under the microscope. So there is a possibility that protein might acquire some kind of stability using the liquid condensation. The way probably nucleosome people are showing that how protein are available when there is DNA damage. That probably liquid condensation is the one of the answer. They somehow probably laminate and ready to work, probably increase the local concentration or ready for work. That's the way probably it is working.
Sir, I have a twofold question. Like, what are the practical applications of the phase-phase transition, and in specifically in case of assays, how we can use that?
No, there is a huge practical application in chemical engineering field or chemical industry. I am not go for that. Even I don't know how people are using. But according to me, that in biology there is a massive applications. So now people able to show artificially you can make liquid condensate inside the nucleus, where you can deliver the drugs through the liquid condensate. Actually, I propose that you can make a polymer, you put the nuclear tag, and you can decide that your polymer where it can go, control the concentration and make a liquid condensate. And that probably you can either tag with your drug or even artificial organelle you want to make. One can do in future, the way field probably is going. Yeah.
Mathematical modeling we're doing in order to move or localizing the polymers we're using.
Mathematical models, there is a called spacer sticker model. I mean, that is kind of mostly famous model that a polymer has to be some kind of spacer and sticker, then every probably interval there has to be sticker so that it can stick. But then again my data is showing this is not the case. Spacer can go and sticker can go. It's all about concentration, all about interactions, and what is the biological context. So if you decrease pH, one protein can form, another protein can dissolve. So if you increase the crowding, one protein might form, another protein might not form. All about critical concentration. That's exactly my data.
Yeah, I suggest that
Yeah.
any further question we can have during the intervals. I want to thank Professor Maji once
Thank
again.